Okay, 107 fans, we are back. Still on Chapter 3, talking about proteins. Still having some issues, but I think I've got it under control. So maybe my allergies will get better once I get all these guys out of my office. So let's talk about monomers and polymers. Remember, mono meaning one, poly meaning many, mer meaning mer. Whatever it is we're talking about. Okay? A molecule, or a macromolecule, is a very large polymer made up of many, many monomers linked together. Right? We have essentially 3 different types of biological molecules. The biological molecules, the big 4, are what? Proteins, nucleic acids, carbs, carbohydrates, and lipids. Woohoo! Right? Fat. Those are the big 4. We're talking about proteins first. They're all macromolecules. They are all polymers. They all are made up of individual units linked together in some way. We need to know what the unit is, what the linkage is called, how the linkage happens, and their overall structure for each one of these guys. We're going to start with the proteins. Okay, when you link amino acids together, each monomer, right? What is the monomer? This is an amino acid. What is an amino acid? NCC. R group. Hydrogens on it. Each one of these is an amino acid. All linked together. Polymerization just means putting monomers together, making a polymer. We need to know what the monomer is and what the bond is called. For amino acids, it's a peptide bond. I'll give you a little foreshadowing. Amino acids linked together through a peptide bond form proteins. Woohoo! Polymerization, the process of putting them all together, requires energy and so therefore is not spontaneous. Right? It's a condensation reaction, also known as a dehydration reaction. Right? Either one, condensation, like when things condense, when water condenses on a window on the inside of your car, it makes water droplets. Condensation reactions release a water molecule. Dehydrate something, remove water. Hydrolysis is the reverse. Right? Break polymers apart by adding a water molecule. We're mostly going to be talking about condensation because that is what drives polymerization. And as it turns out, all the biological molecules use condensation reactions. So you should never, ever, 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 ever get that wrong. Monomer to polymer, no matter whether we're talking about proteins, nucleic acids, lipids, carbohydrates, is always condensation reaction that causes it to go. Why? Because to link 2 amino acids to form this peptide bond, right? NCC, NCC, this carboxyl group and this amino group reacts together. Okay, we need energy to make this happen. When that comes together, if you follow the hydrogens and the oxygens, you will see we lose 2 hydrogens and 1 oxygen in order for this bond to form with all the appropriate shared electrons in those outer shells being full. This is a covalent bond, sharing of electrons. Why did we care about all that biochemistry? Because we need it now. Sharing electrons is a covalent bond. This slide is the semantics. Yes, you have to know peptide bond links amino acids through a condensation reaction. Carboxyl group of one, amino group of the other. Forms this polypeptide. Okay, oligopeptide or peptide is generally fewer than 50 amino acids. It's referred to as a peptide. More than 50 amino acids is referred to as a protein. we can use these really, if you say peptide, it always means a small linked amino acids. Protein, it could be small, it could be large, right? Polypeptide, poly, lots of peptide bonds. Could be any size. Okay, technically, these are the rules here. Okay, but I will try not to use those and say peptide and assume you mean something small, or polypeptide or protein and make you know those. But that's convention, so it's something that should be in your head. The peptide bonds are controlling the backbone. So amino acid, peptide bond, amino acid, peptide bond, amino acid. R group orientation. R group could stick up this way, R group could stick down this way, right? Two together, they would interact, right? Or sticking out the other side, they could interact. Kind of controls where they go. The chemistry controls how those R groups are stuck together. Directionality, the end where the amino group sticks off is called the N terminus, the amino terminus. The end where your carboxy group sticks off is called the C terminus, carboxyl terminus. Peptide bonds also result in flexibility, okay? Single bonds on either side of the peptide bond can rotate. The whole thing is then flexible. So it doesn't have to be in this straight line. It could be, but it could be in any sort of orientation, right? Depending on the R groups and how they interact, right? There's any way that this could be set up. It's very flexible. It's not just in a straight line. I know we think about it really just being in this straight line, but that's not true. Setting it up like this is useful for us because it shows the primary structure or the sequence of amino acids, but that's not necessarily what the shape is. Here's a little picture. Again, which side is the N terminus? Which side is the C terminus? If we asked you that question on an exam and you saw this here and it's an amino group, what is it? N term. What is it? C term. That also tells us that this was the first amino acid added in the polymerization, and this one is the last one. So not only does it give us a direction, it shows us how they were added together in this direction when we built this through polymerization. Here's another picture that was sort of the stick model, right? This is essentially the polypeptide chain just drawn out. NCC, peptide bond, NCC, peptide bond, NCC, peptide bond, peptide bond, peptide bond, peptide bond. Okay? N terminus, N terminus, C terminus, C terminus. If you were just shown this, you'd need to be able to tell me where the peptide bonds are. Okay? It's not this one. No, not at all. This is one amino acid linked to this amino acid, linked to this amino acid. This is peptide. This is peptide, this is peptide, this is peptide, this is peptide. The rest of them are just bonds within the NCC, the individual amino acids. Blah, 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 function follows from structure. Proteins do their function based on their structure, their shape. In Dr. Stein's favorite word, their conformation. That's equals to their shape. If a protein is not shaped correctly, it won't do its job. Specific shapes do specific jobs. If it falls apart, misfolds, any of those things, it will not do the same thing. The shape, the conformation is exceptionally important. Okay, 4 levels of structure. We need to understand these, how they form, what causes them, and why it's important. Primary structure is this sequence, right? Which amino acid linked in what order? The N terminus and the C terminus. Number 1, number 2, number 3, number 4. What are those and what order is it in? Okay. You must have a primary structure in order to get anything else. So the primary structure is the sequence. Does not have, which does in turn affect secondary, tertiary, and quaternary, right? But it's really just the sequence, primary structure, just the order of the amino acids. Okay, the order of amino acids and what they are. In the case where, right, there's a glutamine here, right, this is within the hemoglobin gene, normal red blood cells, normal hemoglobin. We change one amino acid in this giant protein, might have, I don't know how many, 300 amino acids, and that screws up the hemoglobin and causes sickle cell anemia. Okay, the hemoglobin protein is not shaped correctly. In this case over here, it's shaped like a big glob. Over here, it actually goes together. So that is very important. Secondary structure is caused by hydrogen bonding, just H bonds of the backbone, not R group. That's all you have to memorize. Has nothing to do with the R group. It's a carboxyl group and amino group. Okay, hydrogen bonding form as alpha helices and beta pleated sheets. It depends on the primary structure, which amino acids are next to each other. Okay, but nothing to do. Okay, hydrogen bonding between backbone atoms of peptide chains. Right, this is a backbone. Look, NCC. This is the hydrogen. NCC of another, the oxygen. Hydrogen bonding, electronegative, electropositive. Partial negative, partial positive, attractions, causes this kind of shape or this kind of shape. You need to memorize alpha helix, beta pleated sheet, secondary structure of proteins caused by hydrogen bonding in the backbone. Tertiary structure in polypeptides, the third level, is interactions of the R groups. Okay, between R groups, R to R or R to backbone. So always are involved. Secondary structure are never involved. Tertiary are always involved. Could be hydrogen bondings, hydrophobic, van der Waals, covalent disulfide bonds, ionic bonds. Anything to do with the R group being involved. Okay. Interactions between R groups and backbones. Okay. Tertiary structures are very diverse, full of alpha helices, full of beta pleated sheets. Disulfide bridges. Hydrogen bonding between these guys and the R group, or between the hydrogen and negatively charged atoms in the side chains, R group. Tertiary structure. Hydrophobic interactions of the R groups. If it's a hydrophobic R group, it can interact with another hydrophobic R group. Van der Waals forces, weak electrical interactions between hydrophobic side chains, getting together, R and R. Covalent disulfide bonds. If there's a sulfur in the R group, those two sulfurs can interact with each other. Or ionic bonds between groups full and opposing charges. The acidic to the basic, right? A positive charge to a negative charge interactions. All the things, R groups involved. Tertiary structure. Here's a very common disulfide bond. Here's an ionic bond. Okay, van der Waals forces, these are both highly hydrophobic. It brings them together away from the water. Hydrogen bonding between a side chain and a backbone. Hydrogen bonding between two side chains. Causes change in shape depending on the sequence of amino acids, where they are. Secondary structures in the backbone. Primary is the sequence. Tertiary interactions R and R, or R and back. The last level that not all polypeptides have to do is quaternary structure. All must get to the tertiary. All have one primary, secondary, and tertiary structure. Every protein must. That's just what happens. By chemistry, you get to that level. You can't not get to tertiary structure unless you heat the crap out of it and totally denature the protein. That'd be back down to primary. Quaternary structure is when two different proteins get together to form function. Okay, so a dimer, two different proteins. This is a protein, this is a protein. Two different genes, two different RNAs, two different proteins get together to do function. Neither one are functional alone, need to be together. We talked about hemoglobin for sickle cell. It's 4 separate proteins. Alone, none of those can do diddly squat. Together, they carry oxygen and save your life. Woohoo! We love hemoglobin. Okay, so all of these, right, summary. You need to know this to read it to you. What do we need proteins for? Lots of things. All this stuff you think about that cells can do, right? Enzymes. We'll talk a little bit more about enzymes when we get to the enzyme section in glycolysis. So that's going to be all for today. I appreciate you listening, and it's been nice and quiet in here, so I think everybody's sleeping. So I guess I won't actually try to wake up any of the animals. So I'll just say goodbye, and thanks for listening. And I will email you some homework because I completely forgot to set something up. Okay, bye. Oh, that guy's here. Okay, thank you.